Okay, picking up where we left off for the power outage. Sometimes a pathogen can cause leaf senescence. So if a plant realizes there's a pathogen there, it might just shed the whole leaf as a defense mechanism. Leaf senescence is caused by the production of hormones like ethylene and abscisic acid, but mostly ethylene. Ethylene being the same hormone that causes the ripening of fruit, like in banana. Functions other than photosynthesis. Remember, I'm going to talk about photosynthesis in the next class, but leaves do other functions than photosynthesize. They are support structures. Sometimes leaves will actually support whole areas of the plant. They are substitutes for flower petals. They can also trap prey, like Venus flytrap or the pitcher plants. Modified leaf structures include tendrils. So grapes, tendrils will actually be modified leaves, where a cucurbit modified tendril will be a modified stem. So tendrils could be one or the other depending on how the plant developed that way.
What about like a lilikoi vine? Some of these vines that have those things that come out of it.
Modified stem.
Yeah, it's a modified stem.
Yeah. Just like a grape. So you have tendrils for support. In areas where plants become wintered, so in like northern United States, Canada, and all that, we have bud scaling, where leaves will actually, a modified structure will be created from leaf structure that will actually scale over and cover up zones of growth, waiting for winter to come to protect, and then it will senesce those, and then new areas of growth. You have bracts, which are the substituting for flower petals, like in bougainvillea. So you have the very, very small flowers, bougainville, but the colored leaves that look like flowers, which do attract insects, which is the reason why they change color. So it's not an actual leaf of the flower. The flower is very, very, very small on a bougainville. Okay? Succulents, water retention, like cacti. And like aloe and such. And then also defense, spines, like a rose bush. Is it straight or is it not? If it's straight, it's probably a thorn. If it's not straight, it's probably going to be a spine. Okay? And let's move on to roots. The functions of roots, we're going to talk about that. We're going to talk types of underground root systems. We're going to talk about root structures and function, root tissues, root cell growth and specialization, root mineral absorption, sites of selected absorption, and microbial associations. Let's get to it. Root function. Well, they anchor plants. We know that. Hold them to the ground. It's a place where plants store food. They bring sugars from the leaves, bring it down to the roots for storage. Roots are also zones of hormone and secondary compound production. So those communication tools are usually produced in the leaf or in the roots. They can also support stems, like in corn. Look at corn. There's no woodiness to it, but it stands straight up out of the ground. If you look at the roots, it's because the roots come out in a certain way that support the base of the plant. Of course, nutrient absorption, as we all know. The expelling of waste materials. We touched on this on soil fertility. What ion will plants expel?
Chlorine.
Hydrogen?
Yeah, hydrogen. Good job. So plants will expel hydrogen into the environment, changing the pH, and which is a waste material. Also, roots can change micro niches. So where a root hair grows into can create an area of high or low pH, high microbial activity, high moisture. So it can actually change the soil environment to what it needs. Types of underground root systems. Of course, we have taproot systems, like a tree, which has a very central root going straight down as deep as it can go. Adventitious roots like the stolons, they're on the bottom of that table, going across the surface soil. You also have fibrous root systems, like palms or corn, or pretty much any kind of monocot is going to have a fibrous root system. Also, a cutting will have a root system. So if you take a cutting of a, I don't know, like an air layered cutting of a avocado tree, and you put that in the ground, avocado tree is not going to have a taproot like an avocado tree should. It's going to have fibrous roots. And if it gets blown over when it's 20 feet tall, you'll know why. So always use grafted, tap-rooted plants. So have somebody graft something that has a taproot, and then you take the cutting and you graft it on so that your tree has a taproot. Okay, so there's feeder roots and branch roots. Feeder roots are going to be exploratory. They are thin, they are widespread, they will spread usually very close to the surface of the soil. Eventually, those will find nutrient sources, become bigger, fatter, and more established, and then become branch roots. Those branch roots will then send out feeder roots from there. Okay? So, when you pull the soil away or you harvest a crop and all that, those roots that are very close to the soil surface, when you tip it over, those are all branch roots, and those are all feeder roots. Root structure, let's look at you got the root tip, which is the very end of the root. This is the area of growth. This is what an apical meristem, right? Area of growth, root tip. You also have root hairs, microscopic root hairs. So these are hairs on the side of the root that bring in nutrients and water. Root apical meristem. So that's going to be where? In the root tip. Just behind the root tip, because the root tip is a protected area of the root. You also have the root cap, which is a hard shell at the very tip here so that the root can drill into hard surfaces and not get damaged. And you have root border cells, which are going to be the mass-producing cells, the cells that will be changed into more specialized structures. And then you also have a mucigel. It's a lubricant, which is a layer of gel that is excreted from the root on the outside of the root so that it can get around much easier. Root tip regions. Well, let's look at the root tip specifically. You have three regions. You have the region of cell division, you have a region of cell elongation, and a region of maturation. So let's look at cell division. Right here, we have a root cap. Remember I said it's a hard little thing at the end? And then just beyond that, you have that zone of division. This is going to be the apical meristem. Right here, see? Apical meristem. Cell division. And then you have area of cell elongation. Once the cells have established their presence, maybe not specialized yet, but maybe getting those codes to know where it's going to be, it'll start elongating, bringing in water, letting go of a little bit of water, changing the chemical makeup of its cell wall. And so this is where you have elongation establishing itself in the root. And then you also have zone of cell differentiation, and also called zone of maturation.
So roots can actually shrink.
No.
Most zone of cell elongate. Oh, it's just going to elongate.
Yeah, it's going to elongate. Okay. It's not going to generally shrink down because they're already very, very, very small. As those stem cells, those early development cells are really very, very small. They will almost all of them elongate into an established cell. And during that maturation, this is when you get your specialization. You have some of the cells turn into root hair cells, some of them turn into cortex cells, some of them turn into vascular tubes. So nearest the back, That's when we get our zones of maturation. Okay, specialized root structures. We talked about the cortex earlier. This is a root, guys. So we're talking about roots right now. Let's do a cross-section of a root. I just sliced it right across the top. We're looking at it under a microscope, and we have our cortex, which is this area all in here. Remember I said it's spongy? And it's a starch storage area, holding sugars. There's also plenty of airspace between these cells, allowing for gas exchange and water to move between the cells very, very easily. Well, where there's air spaces, there's always going to be water, right? Remember in the soil, 50% of the airspace is taken up by water. It's really no different in the cell structure, or the root structure. And then dissolved minerals are in there as well. So nitrogen, phosphorus, potassium, all those wonderful nutrients that it needs has found its way into this cortex because it's being stored in addition to the sugars waiting to be used. You have the epidermis, which is the outside layer, right? I'm sorry, endodermis. You have the endodermis here, wrapping around the vascular tissue. Innermost cortex layer, it's a waterproof barrier between the cortex and the root core. Waterproof barrier. So this is all storage. It's not going to leak into there. It's only going to bring in what it wants. Waterproof barrier, root core, and then acts as a one-way filter for minerals. So it'll take up all kinds of minerals, but if it only wants copper and beryllium and a little bit of nitrogen, that's what it'll push through. Okay? So it's kind of like a gateway or a gatekeeper.
Well, I have a question on that last one. You say it's a waterproof barrier, then how does the water get into the xylem and the phloem in the center if it's not allowing water to go into there?
Osmosis.
Okay, but osmosis,
Okay.
It's going from there through.
Actually, in osmosis, water doesn't go. It transfers through a semi-permeable layer. What it'll happen is it'll create a zone of need for water on the inside of the vascular tissue. So it'll create an area of lower osmotic pressure than was on the outside in the cortex. And so it will actually percolate it in. So it's not waterproof, but I guess it is waterproof when it wants it to be, you know. It's not going to allow just this constant flow of water coming in. It might just pair the calcium molecule it wants with a water molecule, and then that's how it takes it in, and then it gets rid of the water or puts it into the xylem, and then it takes advantage of that nutrient.
Because the water is coming from the root itself.
It is. This is true. I don't know if waterproof was the right term, but certainly a semi-permeable layer.
Because it needs a lot of water, right? Going
Right.
to the plant.
Right. This is true. I don't know if I would have written waterproof if I saw that again. All right. So you have the primary vascular system. That's in the inside, yeah? That's just on the inside, that little star thing. Primary vascular system has a core. In the core, you have the xylem and phloem. Remember, guys, we're talking about roots here. So just like a stem, it's very similar. It has the xylem and phloem right in the center. It also has a pericycle, which I mentioned earlier is one of those first developing type cells. Site of branch root development. Do I have a photo of this? I don't, but draw a picture. Well, essentially I can just kind of straight it. So we have our pericycle, right? Which is this area around, just on the inside of the endodermis. Remember, there's still cortex on the outside of this, right? So this is just on the inside. This pericycle here. I'll draw it. So we have our core, right? Xylem and phloem, right in the middle. And then we have our pericycle nearest the outside. Kind of in this region, right? And then of course we have all this cortex on the outside. So, what'll happen is, branch root is a root that just grows right out of another root. And the plant will decide, hey, I'm getting chemosensors, I'm getting indication that there's nutrients and water in this region. I don't have a root there yet, I need to send one out. So it'll literally just start growing right there. It'll start developing and start getting bigger. It'll start creating a channel to the xylem and phloem, and eventually a structure will come all the way out and stick out. And then it'll grow and grow and grow. Eventually, this root here absorbs nutrients and water. It gets brought into the vascular system, which then attaches to this vascular system and on up and out. So whenever it wants to, it could have a straight root. It could just start developing a new one and going right. That's why you have all this branching. Okay?
But when you have the root that is coming and coming, things, there are other roots, smaller roots coming out of it.
Right.
That's not that.
That's that.
Oh,
so you have a main taproot that
Right,
goes down,
right.
and then it'll start branching out from the main taproot.
Yeah.
Okay? Or just any other root, this will be created. So when you have very, very fibrous roots, I mean, you got a lot of division, you got a lot of this going on. I think I might have a slide.
Banyan tree or something.
Yeah, so like in a banyan tree, this will happen on the stem, but not on the root. So right on the inside, it'll just decide I'm sending out a root and just poke it right through the stem and straight on down, gravity and pull it. Okay? Specialized root structures include the epidermal root hairs. There's a picture of the epidermis and some microscopic pictures of root hairs. Root hairs are mature epidermal cells. So as the epidermis is developed and changes throughout time, those cells will turn into root hairs. And once they are broken or used up, they break off and are replaced by more epidermal cells, which eventually evolve into root hairs. Okay? They're finger-like outgrowths. They reach water and minerals in soil pores, like on the surface of clay, and all these tiny little niches that roots can't reach into, only root hairs can get in. So these are one cell wide, man. They can get in anywhere. And they do selective transport, in that they will come across all different kinds of nutrients, and it will only absorb the ones it is programmed to absorb at that time, because that's what the plant wants it to do. Mineral absorption. Root xylem contains two nutrient pathways. So nutrients can be absorbed by a root symplastically and apoplastically. Symplastically is water and nutrients that are selectively taken up by root hairs. They transferred to the next cells via the cytoplasm. And remember that plasmodesmata? That's that plasmic exchange, the communication from one cell to the other. I brought that up way in the beginning. And that's how nutrients will go in through the root hair, and the nutrients will be passed from one cell to the next, to the next, to the next, to the next, until they are brought to the vascular system and taken up to the plant. You have apoplastically, which is something more like this. It will skip over the root hair, find the epidermis, find a cell of the epidermis, and the plant will allow that water and nutrients to be absorbed between the cells. And it'll find its way between the cells and eventually into xylem vessels. So you don't necessarily need the root hair to absorb nutrients. Some nutrients can't be absorbed by root hairs and only can be done apoplastically.
So in one plant, it would do both functions?
Yes. Yes. Okay. So roots can absorb nutrients between the cells or straight through the cell wall.
Or both,
Or both, right.
Yeah.
So there's a little bit more apoplasty. Sites of selected absorption. Selected absorption, as I mentioned, is when a plant decides to take up certain nutrients and not others. So this happens at the epidermal cells and at root hairs. So the epidermal cells won't allow in aluminum because it's toxic. It just won't.
Oh, it really won't.
It just won't let it in. But you have another molecule which is the same, about the same size radius, and is plant beneficial. It'll just allow it to go right in. Okay? Selective absorption. We know very little on how that works.
Sorry.
Yeah.
So each root has just one core that has xylem and phloem.
A main root, and then you can have
Main
have
root.
branching roots from
So
there.
like a big tree has this giant root. It only has that one central thing going into the trunk.
It has the taproot that's going down to groundwater, and then from that taproot there are branched roots coming out from there.
So each one has their own central core, phloem, and
Yes,
xylem.
everyone has its own little tube, all its own structure is just like this.
And they all converge.
Converge at the
yeah,
taproot and go on up,
and go
correct.
up, so that you have the same amount of cells going up. I mean, not cells going up, but pathways going
Yeah,
up
right.
as you have roots.
Yes, true. This is true. Okay?
What do you say? Oh, I just wanted to get that.
He got it. Sites of selective absorption, as I mentioned. So it's basically like a screening of mineral content of the water. How does it screen it? What if I said it started with a C and ended with an arge? Charge. So, because of charge, it'll send out a certain charge. It'll be able to absorb a 2+ ion as opposed to a 3+ ion or whatever. Okay? So, it can sequester aluminum and other minerals. A root can create organic acids. They do it all the time. Main component of the organic acids is hydrogen. So, it creates a organic acid. If it finds an incredible amount of aluminum, which is toxic to it near the root zone, it'll create an organic acid which will bond to the aluminum molecule and make it so that it can't be taken up by plants. It'll actually take the thing that'll make it sick and make it so that it can't absorb it. Dissolve mineral bonds. So a plant can also excrete these organic acids into the soil environment, which release nutrients from the parent material. Secrete transporter proteins. These are great. These are proteins that are oozed out of the areas of absorption of the root into the soil solution. These transporters have a specific charge. It will send it out into the soil solution to grab onto nutrients that the plant knows it wants, creating a different charge, and then it will send out an opposite charge, hoping to grab a hold of that nutrient. So it'll send a protein out which grabs onto a nutrient, and then it uses the change in charge to suck it back in. It's like fishing. Just kind of sending a cast out there and then bringing the nutrient back. Sending it out, bringing the nutrient back. Microbial associations with roots. We're going to talk a lot about this, guys, in the next couple classes. Mycorrhizal relationships. These are fungi that have relationships with roots, where there is a symbiotic mutualistic relationship where the fungi will find nutrients and water in the environment that is further away from the roots that the plant could reach, bring those nutrients to the plant and trade it for sugars. It eats sugar. It doesn't eat nitrogen, phosphorus, potassium, but it's more than happy to go gather some and trade it for those sugars that it wants because it has the ability to do so. There's also nitrogen-fixing bacteria. Some of them are free living in the soil, But when I'm talking about associations, we're talking about legumes that produce special structures in their roots that house bacteria that in anaerobic conditions without air can take nitrogen gas from the atmosphere, which is absorbed through the plant, brought into the plant structure, brought down to these bacteria in these special structures in the roots, and then that microorganism is able to break the triple bond that is the nitrogen gas. Very, very hard to break that bond. We have only been able to do it since 1946. And bacteria are able to do it pretty readily. Okay? And so that is nitrogen fixing bacteria. It takes nitrogen gas, turns it into plant uptakeable nitrogen, gives it back to the plant in exchange for sugar.
So what would be like names of bacteria like that? Or is it what?
Rhizobic bacteria. So there's special names for rhizobic bacteria. There are special bacteria that are found in the environment that plants will either absorb, but most times than not, when you're talking about nitrogen fixing bacteria, this bacteria is inoculated into the plant by the mother. The mother will have that bacteria in it, and then when it makes the seeds, it will inoculate the seeds with the bacteria, so when the plant grows, it already has the bacteria ready to fix the nitrogen for it.
So it's not something you can bring or add to your soil.
It is. You can inoculate it. We can inoculate it with certain microorganisms, and it actually is pretty readily available in the soil. So there's a chance, like when I grow sun hemp, I've never had to inoculate because the soils have enough organic material.
Because the sun hemp will create that.
Yeah, sun hemp is a legume, and it will create special
okay.
structures. Okay? There's also enzymatic release of nutrients. Well, as microorganisms break down soil and organic material, they're going to have a direct effect on the nutrition of plants by releasing nutrients to the plant. So that's definitely a direct microbial association. Most of the nutrients a plant absorbs, if not all of that, including when you put out fertilizers, is absorbed through enzymatic release of nutrients from microorganisms. So when you put out a fertilizer, 9 times out of 10, that fertilizer needs to be converted by microorganisms into a plant uptakeable form. So that's what we're talking about. Plant reproduction. Asexual reproduction. Plants will reproduce asexually without intercourse, without the fun stuff, and just do it alone. They also do sexual reproduction, which is in this table here. Where do we start?
We're going to end it the same way.
The embryo, yeah?
Yeah.
Best way is probably to always start at the embryo. Embryo being fruit, right? Fruit, or the seed that's in the fruit, more specifically. So as the seed comes out and grows, it becomes a plant, a gametophyte. Remember, what are gametes for us? Sperm and an egg. What are gametes for plants? It's the actual plant that we see. So where their life cycle is in that phase, ours is sperm and egg. The gamete creates sporangium during the sporophyte phase. Meiosis being the breaking apart of chromosomes. Chromosomes are separated from one spore which has 4, right? 4 cells in it, each one having chromosomes, into single cells having 1/4 less than the 4 put together. So 4 come out, and then you have 4 different ones, usually 2 female, 2 male, or whatever the plant dictated, right? Spores come out, create gametophytes. Either a spore will come out and make a male plant. Male plant, or a female plant. All plants are either male or female. Very rarely are they both. sometimes plants can have flowers and be a male. They can have ovules and be a male. It depends on what you do to that plant. You give the right hormones to a plant, you can change it from having flowers and not having flowers. It does not change it from being a male to female.
Really?
So if I take a male plant and I give it the right hormones to change it to a female structures, flowers, as opposed to creating just spores, create flowers, I can do that, the right hormones, and then when those flowers make seed, or make fruit and make seed, the genetic code that is on those seeds is still male. So every one of those that comes out of that plant will be a male, even though it came from a plant with flowers.
Wow.
The structure, the sexual structure, does not have anything to do with the genetics of the plant. This is why we've been able to play with plants like that for a very long time. Okay? So, you have a male plant and a female plant. The female plant makes archegonia, which is the egg, and sperm, or the antherium and theridium make sperm. They come together in the ovule, right? So the egg is part of the ovule of the archegonia. The sperm goes inside the ovule, fertilization, and we have a seed. This is how it works, back and forth. Okay? That is sexual reproduction.
It's like in a vanilla bean,
plants.
you have to put to make it happen.
Yeah, we have to fertilize it,
Right.
yeah. A lot of plants are like that. Sexual plant reproduction. It is the fusion of a male and female gamete. I'd already mentioned it. And a gamete is, once again, single cells produced by an adult which are specialized for reproduction. What are our gametes? Sperm and egg. Sperm are male gametes and eggs are female gametes. Sexual plant reproduction, the fusion of gametes to form a zygote. I think I'm repeating where we were 2 hours ago, but we're going to get pretty deep into it, so I want to keep doing it. Fusion of gametes to form a zygote. Gametes come together, single cell come together to make a multicellular creature that will become an adult, which can reproduce. Meiosis, right here. The definition is a nuclear division process that reduces the number of chromosomes by half. So if you have 4 cells, it gets chopped into 4, each one of those gets cut in half. Watch the video. Hopefully the video will make sense to you guys, and hopefully we have enough time to watch it. So, sexual reproduction, what are those on the right? Those are spores. Ensure genetic variability. That's why plants go through sexual reproduction. Without genetic variability, you get some pretty stupid plants, right? Well, you have plants that are not adapted to changes in the environment. Okay, because that's the key to having genetic variability, is actually having a gene that is adapted to the environment. Uses a lot of energy to do sexual reproduction. Regardless of this, most plants do it, and they do it a lot. Think about how many spores are out there and how much pollen is out there, and we're talking about fruits and all that stuff. I mean, most plants are going to engage. Of course, we have asexual plant reproduction as well. I lost my picture there too. Requires fewer resources. It's quicker and more efficient than sexual reproduction. The problem is there's no genetic variation. So there are whole patches out here in Hawaii where you have one plant, invasive plant that landed there and sent off another clone, and the whole entire hillside is all genetically the same plant. Okay?
Like Myconium.
Only requires one parent. All progeny are genetically the same. And it happens a lot in areas that are harsh environments. So arid and cold environments, you have a lot of plants where there's not a lot of pollinators. You're going to find plants that typically do asexual propagation, like cactus does a lot of that, and a lot of the conifer trees and stuff like that being genetically the same. Some whole regions up in Canada, forests are the same tree. They're all the same, genetically the same tree, okay? As compared to other life cycles, comparing it, of course, to animals and some algae, animals are usually gametic. Well, they all exist in the gametic phase.
What does that mean?
Means male or female, or, well, gametic, meaning that we are gametes. We are the result of gametes. That's what it means. So a sperm and an egg came together, created a zygote. That is a gametic existence.
Like evolution?
Well, no. It's just vocabulary, I guess. Okay. Gametic existence is being the result of two gametes coming together and creating a zygote. Sometimes, some creatures in nature stay in the zygotic phase, and that would be like protists, protista. So these will be very similar to bacteria. They do not leave the zygotic phase until they go into reproduction, and very quickly will go into a sporic phase, create the spore, and then go back to the zygotic phase. Sporic phases, like land plants, exist as you see them, are in the sporidic phase. They are the results of spores being released into the environment. Spores being
Hapu and Ohia.
all right, let's watch some videos then. Okay. We're going to start by looking at the fertilization of mosses. Remember I said mosses were the first plants really to establish themselves terrestrially, and they do not have stems because they haven't evolved yet, but they have different life cycles in that compared to plants. So let's take a look here. We might go 15 minutes over today. Is that okay, guys? Chalk it up to the power.
Forests are dominated by large plants that crowd one another in their competition for light. But far below on the forest floor, there is a group of tiny plants known as the mosses. With over 10,000 species, these plants thrive in many habitats. These moss plants form gametes and are therefore gametophytes. Moss gametophytes are haploid, containing only one set of chromosomes.
Most gametophytes are haploid. One set of chromosomes, like sperm and an egg, right?
Their small and compact form enables the stems to stay upright, while rhizoids anchor them to the soil. Moisture easily accumulates around the densely spaced stems. This often leaves a film of water on the leaves and on the stem tip. Hair-like growths called paraphyses help to hold the water around the stem tips.
How does it hold the water there? Adhesion. Right? Water wants to
that's
stick to a
adhesion,
surface.
right.
So if it has all these rods just sticking out of the top, it'll just hold a little ball of water.
Some stem tips are male and contain reproductive structures called antheridia. These are complex organs consisting of many cells. Each is supported by a stalk.
It's on the inside of the cup.
The upper portion consists of an outer protective jacket that surrounds a group of sperm cells. At the tip of a female plant, there are numerous reproductive structures known as archegonia. The top of each consists of a slender neck. The middle region contains a chamber called the venter.
Don't go crazy on the vocabulary here. Just the
The
concept.
base of the archegonium is attached to the moss stem by a stalk.
These are bryophytes. We
An
don't
egg
really
cell
deal
is
with them so
formed
much.
inside the venter. Inside the neck, the breakdown of cells forms a central canal containing sperm attractant. A rain shower provides the ideal conditions for sperm release and their transfer to a female plant. Droplets of rainwater collect at the tips of the moss stems. Inside the drop of water at the tip of the male stem, the antheridia open. This releases cells containing sperm, which float up to the surface. Raindrops splash some of this water out of the male plant. Some splash drops contain sperm cells and fall onto the tip of a female stem.
That's it.
The sperm now escape from their surrounding membrane and use their flagella to move about in search of an egg.
Is that really the real thing? The
The
pictures you see,
archegonium
is
now
that the real
opens and
sperm?
releases a sperm attractant.
Well, what they look like, yeah, graphic
Oh, okay.
The
interpretation.
sperm swim towards the source of the attractant.
I'm pretty sure
and
they
then
don't make
feel-
squeaky noises, you know.
Once inside, they are guided down to the egg.
Yay!
I'm getting dizzy.
Yeah.
The first sperm to arrive enters the egg cell. Fertilization is completed when the sperm and egg nuclei fuse, creating a diploid zygote.
Diploid. 2 sets of chromosomes, right?
After fertilization, many changes take place on the female stem tip. At first, the zygote remains within the venter, where it forms an embryo. However, one end of the embryo soon grows out of the venter and into the female stem. This allows it to obtain water and nutrients from the female plant. The other end of the embryo grows upward. The venter expands to accommodate this growth, but eventually it is split in half. The embryo now forms a very long stalk or seta that lifts the top half of the venter up into the air. This results in a small diploid plant, the sporophyte, attached to the tip of the female stem.
Next time you see mosses, and you'll see this structure sticking out of the top,
Uh-huh.
and you'll realize that is a different life
Right.
stage for the plant. It's not going to look like the same thing anymore. So that's 2 sets of chromosomes
These
together.
female stems are
Just like
normally
us.
fertilized
This is an
and
us.
supports
The version of us.
sporophytes above them.
And look at how fast it goes away. It grows up, releases its spores,
The
and
tip of
dies.
each is covered by the torn venter, or calyptera. This soon falls away, exposing a capsule or sporangium. The capsule contains fertile tissue consisting of sporocytes.
Haploids. 1 set of chromosomes.
Inside the nucleus of each sporocyte, there are 2 sets of chromosomes. The nucleus divides by the process of meiosis, forming 4 haploid nuclei.
So 2 diploids just turned into 4 haploids. Haploid meaning 1 set of chromosomes. 2
Oh.
diploids is a 4, right? Just got cut into 4 pieces. That's meiosis.
Thin walls now form around each nucleus, resulting in a cluster of 4 cells, known as the tetrad. A deposit of sporopollen produces cells with thick, resistant walls, called spores.
So those crazy shapes on the outside of the spores is a result of that
As the spores are forming,
that
the
shell
capsule
being
dries
formed
out
as
and
it
hardens.
develops.
Soon the lid door, or operculum, falls away, revealing a ring of fine teeth called the peristome. These surround an opening in the capsule. As they dry out, they bend backwards, allowing the spores to escape. A light breeze is all it takes to carry the spores away from the parent plant. Spores carried by the wind may land on an exposed surface. After rain, the spore cell absorbs water and germinates. But instead of growing into a new moss plant, it forms a branch filamentous protonema.
Kind of like a stole.
However, after a period of growth, some of the protonema form buds.
It's like a space station.
Each bud will proceed to grow into a new leafy gametophyte.
And the whole thing starts over again. I'm going to cut it off a little bit early so, we can
these
get out of
things
here.
like that are the stems now? Or is that the roots?
Those are the gametophytes.
The gametophytes.
There are no roots, they're just
Not yet.
rhizomes
We're
there.
not there yet.
Let's look at ferns. So, a little bit more advanced, more plant-like. I knew I should have started this earlier. We're gonna go a little over, guys. Sorry. Wait a minute. That was flowers.
They're pretty.
Wait, that's the same thing. Hold on a second. Oh, that's because I have the same one on both.
Oh,
Give me
fertilization,
1 second.
flowering plant. That's fine.
I got this. Mosses. Here it is.
Sorry, fast words.
Is often been used in artworks such as the koru. Ferns are popular in our parks and gardens, and even brought indoors to enhance our living spaces.
Yeah. Reproduction.
In the wild, ferns are successful at reproducing and spreading throughout the forest. They do this by producing spores. The familiar fern is known as a sporophyte. If a fern frond is turned over, small circular areas may be seen. Each of these areas is called a sorus. In some ferns, the sorus is unprotected. In many others, it is covered by a cap, supported by a central stalk. This protective covering is called an indusium. In some ferns, this covering is cup-shaped. The sorus itself contains numerous sac-like structures. Each of these is called a sporangium. The outer wall of the sporangium consists of a layer of protective jacket cells. As the sporangium matures, a row of jacket cells enlarge to form a band known as the annulus.
This is so interesting.
The outer wall of each annular cell is very thin and delicate. On the opposite side of the sporangium, several delicate lip cells form. Below the jacket, there are two layers of cells called the tapetum, which nourish the fertile tissue within.
Like a yolk.
The fertile tissue consists of sporocyte cells. Each sporocyte cell is diploid, containing two sets of chromosomes, one from each parent. As each cell matures, its nucleus divides twice by the process of meiosis. Each daughter nucleus.
So you see, it starts off opposite of the other one, right? Because this is going to have male and female, right?
Mm-hmm.
In one unit versus the male and female stalks of the bryophytes.
Oh, okay.
Now contains one set of chromosomes.
So it's almost
The
opposite.
cytoplasm of the sporocyte undergoes cleavage. This results in a cluster of four adhering cells, called a tetrad.
But
The tapetum now begins to break down.
everything breaks into 4 cells. Is it any plant? Like in the
Most
production?
spores are going to have 4
Spores.
cells.
Okay.
Depositing a very tough protective coat of sporopollenin. Surrounded by this thick resistant wall, each cell is now called a spore. The sporangia are now ready to open. Opening is accomplished by the annulus that encircles the sporangium. The annulus begins to dry out as water evaporates from its surface. The tension or pull between the remaining water molecules and the wall now increases. The thick inner and side walls resist this, but the thin outer walls are easily pulled inward. Pulling in the outer walls causes the annulus to contract much like an accordion. Shortening the annulus tears the lip cells apart. As the annulus continues to shorten, the tear enlarges and the spore case opens further. When too much water is lost, the water molecules are no longer able to hold together. When this happens, the pull on the wall is released, and the annulus springs forward, closing the sporangium so fast that the spores are thrown out. This process is repeated in thousands of sporangia on a leaf, so that large quantities of spores are released.
Gametes.
When spores land on a moist surface,
Right?
those
Like our
which
sperm and
germinate
egg.
first will form bisexual or hermaphroditic individuals. A rhizoid emerges and attaches.
That's the key, is the hermaphrodicity of that gametophyte. So the gametophyte comes out just like a sperm and egg, right? But the sperm is male and female, one at each end.
The spore to the soil. This is followed by a sheet of cells, which is the young gametophyte or prothallus. A notch forms on the
If you go out to your fern forest, look closely at the ground. These will be on the ground about the size of your pinky nail.
prothallus that contains dividing cells. This is called the notch meristem. Continued growth results in a heart-shaped bisexual gametophyte. The lower surface is firmly attached to the soil by numerous rhizoids.
Got it, right?
Male gametangia, called antheridia, are formed at the posterior end of the gametophyte. The outer wall of an antheridium consists of ring cells and a capsule, which surround fertile tissue.
So the same as the bryophytes, antheridium is the male reproductive structure.
And that's still in the fern? Or that's anywhere now? Where are we?
This is the gametic phase of the fern.
Okay.
At the anterior end of the prothallus, close to the notch, there are female gametangia called archegonia.
Same name.
An archegonium consists of a neck containing a neck canal cell. At the base of the neck, there is a swollen region called the venter that contains an egg cell. Gametangia of both sexes may be present at the same time or at different times.
So the gametophyte, the gamete, came from a plant that had male and female qualities, right? So does the gamete itself. It has both of those sexes in it, being hermaphrodite. So in the gametic phase, it's hermaphrodite, it's going to be able to fertilize itself, right?
The timing of their appearance will determine if there will be self-fertilization or cross-fertilization.
Like, taking back what I just said, it's not going to typically inseminate itself. It just said that, you know, it's going to release the archegonia,
Some ferns,
and then if some other ferns are around there, they sense the archegonia is open, they will then send out the antheridia.
And when flooded with water, a hormone called antheridiogen is released.
So, prevents self.
This hormone will stimulate
Reproduction.
adjacent plants to stop growing and form antheridia, but no archegonia. A bisexual gametophyte in a female phase may therefore be surrounded by several male gametophytes, increasing the chances of cross-fertilization. Being close to the soil, the small gametophytes are easily flooded when it rains. This water plays an important role in fertilization. Water stimulates the cap cell on the antheridium to open, releasing the sperm cells. The flagellated sperm are now able to move within the water in search of an egg.
The noises they make, like, yay!
Sperm consists of a spiral cell body bearing numerous flagella that move it forward. Water also stimulates the archegonium to open. The contents of the neck canal then diffuse into the surrounding water, where they act as a sperm attractant. The attractant stimulates a sperm to swim towards the open archegonium. It then swims into the opening and moves up the neck canal towards the egg cell. Fertilization is accomplished when the egg and sperm nuclei fuse. This creates a single diploid cell, the zygote.
Okay, that whole time was a gametic phase, right? We're now in the zygotic phase. Two gametes coming together. Just like us, we're zygotes. This is the human, phase. Watch how fast it is.
The fertilized egg remains attached to the gametophyte. Inside the archegonium on the lower surface of the gametophyte, the zygote begins to divide, forming the embryo's sporophyte plant. The uppermost part of the embryo, the foot, absorbs nutrients from the surrounding gametophyte tissue. As the embryo grows, the venter tissue stretches and eventually ruptures. A root now grows into the soil for support and to absorb water. Other parts of the embryo form the first leaf and the future stem, the rhizome. The leaf grows through the notch in the gametophyte to reach the sunlight. Eventually, the rhizome emerges and provides additional leaves and roots. In this way, a new plant is formed that matures into another spore-producing fern, completing the reproductive cycle.
So when you see those rhizomes going all the way, or those stolons going all the way out to the base, you see the branches coming up and uncurling, you just now understand where that's all coming
from. The large familiar spore plant, or sporophyte.
I find it to be amazing. Yeah. And then we're getting into the fertilization of flowers, and we're done for the day.
Flowers are a source of pleasure within the plant. It's for this reason the larger, simpler flowers of the many monocotyledon flower by a filament.
Here we go.
Which are the flowers of the many monocotyledons. The small number and large size of their flower parts allows us to easily illustrate their structure.
Almost all flowers have these same structures in different
Each flower has 3 sepals, which overlap 3 petals. Inside the petals there are 6 stamens that produce pollen. Each stamen is attached to the flower by a filament. The enlarged sac at the tip of the filament is called the anther. Inside the anther there are 4 microsporangia. Each microsporangium is nourished by a layer of surrounding cells called the tapetum.
Notice a pattern in a lot of these terms.
A microsporangium consists of diploid sporocyte cells.
Diploid. 2 sets of chromosomes.
For the flowers?
The cellulose
The
walls
spores.
of these cells are soon replaced by a layer of callose.
So their spores have 2 sets of chromosomes.
Each sporocyte now divides twice by meiosis. This creates 4 haploid microspore cells, each with 1 set of chromosomes. This group of adhering microspores is known as a tetrad. Soon the callose is removed and sporopollen begins to form on the surface of each cell.
See how they were all bunched together? Now they've each got their own shell.
That's
Eventually they're going to break off to be their own spore. So single chromosomes in there.
The surrounding tapetum now releases additional sporopollen. One area of each wall, the sulcus, remains smooth, receiving very little sporopollen. However, the rest of the wall is covered in a thick, ornate coating of this tough, resistant material. Each microspore nucleus now divides by mitosis. The lower generative cell separates from the wall and moves into the cytoplasm of the larger tube cell. This is the immature male and is known as a pollen grain. Before it is released, the pollen grain accumulates.
So that same genetic code, right, was in one unit and it split into two, right? So now this is where the relationships between plants and animals you start to see where we have very similar things going on in our embryo.
Nutrient reserves. Finally, most of the water is lost, and the pollen grain enters into a resting state. Before the anther releases the pollen, the cells beneath the epidermis develop thickened walls. This is called the endothecium.
It's like a shell on the outside of all of them, just below the skin.
When the pollen and the anther mature and dry out, the endothecium shrinks. This tears open the anther between each pair of microsporangia, allowing the pollen to escape.
You ever go up to like a lily plant and you touch it one day and it doesn't leave anything on your hands, and you go out the next day and you barely touch it, it's a poof, poof everywhere, right? So that's the during the opening.
At the center of the flower, there are 3 carpels. At the base of each
All
carpel,
ovules have
there is
3
an
carpels.
enlarged ovary. A slender style extends from the ovary. And at the tip of the style, there is a stigma. The 3 carpels are fused together, forming one stigma and a style with a central canal. The base of the canal leads into each of the 3 ovaries. In the young carpels, small outgrowths appear on the ovary wall. Each of these is a megasporangium. 2 layers of cells, the integuments, grow over the megasporangium. A small pore, the micropyle, is the only area that remains uncovered by the integuments. This entire structure is called an ovule. Inside the sporangium, there is a single diploid sporocyte cell. The diploid sporocyte divides by meiosis to form 4 haploid.
A diploid sporocyte. So that's unlike the ferns and the bryophytes. So the spores typically are going to not have both genetic traits in it on the others. This is the difference between flowering plants.
Nuclei, each with 1 set of chromosomes. In some plants, such as Lilium, walls are not formed around these nuclei. But in the majority of flowering plants, walls are formed around each nucleus, resulting in 4 megaspore cells. 3 of these then degenerate.
Why is that?
Evolution.
Oh.
The surviving spore enlarges, and the nucleus divides 3 times, yielding 8 haploid nuclei.
Now we got
2 of these
8
nuclei
single
move
chromosomes
towards the
shown.
center.
And they're all identical.
These are known as the polar nuclei.
Half of them are
7 cells
because it's a
now
result of
formed.
male and female
Oh, okay.
fertilization.
At the micropylar end, there is an egg cell and 2 synergid cells. In the middle, there is a central cell containing the 2 polar nuclei. Above this, the remaining nuclei form 3 antipodal cells. This is the female gametophyte, also known as the embryo sac.
So each one of those, after it split off, became one of those structures within the embryo. So one became the egg, the other ones became the structures and the protective structures around. Very similar
This is contained within the megasporangium.
in human beings, where one of the sets becomes the yolk sac and one becomes the baby. Very similar, right?
Which is covered by the integuments of the ovule. The ovule, in turn, is surrounded by the carpel. The transfer of pollen to the receptor surface of the stigma is called pollination. This happens by various means, such as wind, insects, or other animals. The pollen grain now absorbs water and begins to swell. A pollen tube soon emerges containing the tube nucleus and the generative cell. The generative cell divides to form 2 sperm cells. In order to reach the ovaries, the pollen tube must now carry the sperm down the full length of the canal. It does this by first growing through the stigma to reach the surface of the canal directly below. Here it grows along the moist transmitting tissue, which serves as a path to the ovary.
This actually happens.
Pollen tubes have a very fast rate of growth, often several micrometers per minute.
Wow.
This growth is localized at its tip.
Every time you have a fruit. But it happens 3 times per
Vesicles containing wall material accumulate at the tip, where they are required for wall growth. Behind this, the cytoplasm flows around the cell in regular streams. This is known as cytoplasmic streaming. The 2 sperm cells and tube nucleus continually move forward as the cell grows. Plugs of wall material called callos form, which keep the cytoplasm close to the growing tip.
So, it doesn't need to fill the whole tube with cytoplasm and nutrients and all those things. It just needs to get it to the end, and then it sets up these little plugs along the way, keeping that material up near the front. Like, the goal is to get the sperm to the end. It's not to bring them back. Just get them there.
When the pollen tube reaches the base of the style, it grows into one of the 3 ovaries. Inside the ovary, the tube locates the 2 rows of ovules. Approaching an ovule, the pollen tube grows into the micropyle, where it penetrates the megasporangium. It then grows into one of the synegid cells, releasing the two sperm. One sperm fertilizes the egg cell, forming a diploid zygote cell. The second sperm fuses with the two polar nuclei, forming a primary endosperm cell.
Nutrient source, egg sac.
This double fertilization is characteristic of all angiosperms. The ovary contains hundreds of ovules, and each requires a pollen tube to be fertilized.
Look at all those.
To accomplish this, the style supports the growth of a large number of pollen tubes.
So it's not just one at a time. They just come flowing down that tube.
Mass production.
These grow down the style and enter each of the three ovaries.
And then they have a reggae party.
When the pollen tubes enter an ovary, they each locate and fertilize an ovule.
Like actually somebody saw this is happening.
Doesn't that blow your mind that
Yeah.
we know this is happening because we observed it?
Each ovary now has many fertilized ovules, which will develop into seeds.
A long time ago, too.
Yeah.
How do they observe through a microscope?
Yeah.
But you'd have to watch it over a long period of time, right?
Well, I mean, it's really just it's
After the
experimenting
ovules
with
are
it
fertilized,
more than
the
anything.
flower undergoes several changes.
Because before
Many
microscopes
parts
and
begin to
labs and all that, it was just
Eventually,
experimentation,
these parts
experimentation,
are shown.
experimentation, till you get the same results over and over and over again. That is
Only
science
the
to
ovary
the
remains
hardest level.
healthy and firmly attached to the stem.
That's how we did it for 10,000 years before computers and machines.
Inside the ovary, the ovules are now about to undergo many changes. Each ovule contains two fertilized cells. The nucleus of the primary endosperm cell begins to divide to form endosperm tissue with no cell walls. The zygote divides, forming a large basal cell and a small apical cell. The basal cell produces a chain of cells called a suspensor, and the apical cell gives rise to an embryo. The suspensor now begins to transport nutrients into the young embryo. In monocotyledons, a single seed leaf or cotyledon forms.
You asked me what monocot and dicot means? That's what it means. So you'll have one cotyledon versus two cotyledons. Dichotic, eudichotic versus monodicot. So when a seed comes out, you'll actually see cotyledons, leaves. You have one leaf for the grasses, and you have two for dicot.
As it enlarges, the embryo also receives nutrients directly from the surrounding endosperm. During this time, walls slowly form within the endosperm tissue. A tiny shoot tip forms on one side of the embryo.
This is all still inside the plant.
Below this, there is a stem called the hypocotyl. At the base of the hypocotyl, there is a root. This is covered by a protective root cap. In most monocotyledons, nutrients now begin to accumulate within the endosperm. When nutrient accumulation is complete, much of the water is removed, and the embryo enters a state of dormancy.
Dried corn kernel. Right? Monocot.
However, a different sequence of events takes place in the ovules of most dicotyledons. In these ovules, the growing seed leaves accumulate nutrients and absorb all of the endosperm. As both types of ovules mature, the integuments develop into a tough, protective seed coat. The ovule is now a seed. At the same time that the ovule is changing into a seed, the developing embryo is releasing hormones. These stimulate the ovary to expand and make room for the enlarging ovules. In this way, the ovary wall expands and is transformed into a fruit. In most lilies, the fruit is a tough, dry capsule that tears open to release the seed. The seeds are soon dispersed and will germinate to form new plants, thus completing the life cycle. We have seen that flowers capture pollen and ensure the success of fertilization.
Okay, that's the life cycle of flowering plants. See where evolution comes into play? Plants, or bryophytes become ferns, become seeded plants. And I had a 10-minute crash course, which is a great summary that this guy does of everything I just talked about. So when I send you the PDF, please watch this. It's a great summary. Okay. It goes on for about 8 minutes, but he talks really fast, and his show is called Crash Course. And really, what he does is he talks a million miles an hour about things that you just learned about, and he reiterates everything you just learned. It's a great thing on YouTube. Okay. So check that out. Next class, we're doing plant nutrition. That's Wednesday. And then after that, we got introduction to microorganisms. I'm not going to write that one until I know I got 6 people in the class. So if you want to get in that class, sign up. And then let me know so I can get plenty of time to work on these things. Okay? Soil microorganisms right after that. Remember, that's going to be almost strictly decomposition and breakdown of organic material by microorganisms, releasing nutrients to plants. Plant-associated microorganisms. These are the mycorrhizae, the rhizobia. I'm going to go really heavily into the nitrogen fixation and all the things that I talked about today. And then introduction to entomology and nematology. That's going to be bugs and nematodes. We're going to identify mouth parts and different structures of these animals. Realize that not all bugs are bad, and you learn how to address issues based upon the mouth parts of your insect. These classes are probably, don't quote me on this, but might be 2 to 3 hours long. I doubt I would be pushing 4 hours for these 3. It's not a lot of material there. I'd be repeating myself at some point. And then introduction to entomology and nematology, expect at least probably 4 hours for that class.
So which one you said you're gonna have to consider the amount of people?
4:30.
It's any of these, yeah. I mean, if I can get some of the natural farming folks, they might sign up for something like that. But from what I've gathered, there's not all that much interest in that as there is in plant physiology, morphology, plant nutrition, soil nutrition, all that stuff. But who knows? If I have 6 in the room, I'll rock it out. Did you learn something today? Maybe? Did I bore you to death?
I know what the inside of my eyelids look like.
I only lost you twice today, which is a pretty good run.
But I recovered, man.
I love calling him out. And the answer is, he just wakes
A lot of
up.
information there.
Any questions about what we talked about today? Did I do it right? Okay. Let me know on the survey. Please fill out the survey. I am going to email everybody in the room this PowerPoint presentation right now.
Right.
Okay.
Okay? And I'm going to do PDF probably best, or do you want a PowerPoint presentation? PDF is probably best.
You see that one about the interpreter? I should send you that one about the interpreter.
A brain can handle it, right?
Yeah, you do. Yeah. What's that?
No, like, I mean,
15 minutes.
Okay, I got Gino's on there.
Hey.
Ed, I got you. Boom, you're on. Do I have yours on here?
James, as soon as
No.
he going home, he going Craigslist?
Right here. Come over here and put it in. It'll probably be
Send it to
faster.
Craigslist.
Yeah, just put in your email address real quick. Sorry about my cords here.
Thanks, Joe.
That's it?
Yeah.
Okay.
Cool, yeah. You know what about it?
Who else?
Every day I go to camp.
Go over here and type it in. Unless you just, I can send it to him and he can send it to you.
Yeah.
Okay. So Ed.
Papa sent me yours.
P-I-P-I-K-I,
P-I-D-I-D-I-K-I
5-3.
P.
The M-I-S-S-I-P-I.
P-I-P-I. Ah.
P-P-K,
Ah. P-I-P-I-K-I
5-3
53
at AOL.
at AOL. Anybody else? Everybody else is on here? Okay. Send. Invalid. You guys have invalid email addresses.
Mine is invalid? No way.
No. Do you want to send anyway? I'm sending anyway. Okay. You guys, let me give you a card of mine with my email address. So if you don't have it when you get home, send me one and say, hey, I didn't get it. And then I'll have your email address and I'll just attach and send it back. Okay?
I've got yours.
You should have it in your inbox.
Yeah, I have your email address.
What is it?
No, I'll handle it. Don't worry.
Inbox?
You should have, in your inbox under email.
Okay,
If you don't have it, let me know.
See you guys Wednesday, yeah?
Room?
yes.
No, the soil didn't get ruined. I actually didn't end up using it. The IMO3, the mill run and stuff
Yeah.
that I was doing, it got put together and then it went up to 160 degrees and I didn't have anybody there to turn it. And I showed up like a couple days later and it was like